Meiosis and sexual reproduction. Let's do a lecture. Alright. So, we discussed this previously, the concept of being diploid and haploid. You, again, as a human, have 23 pair of chromosomes. 23 pairs. That would be 23 from your mom via an egg, and 23 from your dad via a sperm, coming together to unify into 23 pairs of chromosomes, or what we'd call 46 total. Okay? Now, that is a diploid number. 23 pairs is a diploid number. Your haploid number would be 23 total chromosomes. Not 23 pair. The haploid number is 23 total. So the idea is this. Mitosis forms, or rather takes a single cell and makes two identical cells from that initial cell. This is taking a single 2N or diploid cell and making two identical 2N or diploid cells. This is how you were initially as a fertilized egg, and then you grew and grew and grew, and growth and development and getting taller and what have you. It's all mitosis. Alright, it's all mitosis. If you get a cut on your finger today, the cells there will divide mitotically to fill the gap where the cut is, making diploid, mitotically formed cells. As opposed to meiosis. Meiosis is where we take a single diploid cell, as we've been talking about this whole time, and you double its DNA content, and then split them all in half. This is meiosis 1, and then this is meiosis 2. The idea is that in meiosis, for sexual reproduction only, you take one diploid cell and you make 1, 2, 3, 4 haploid cells. Those could be sperm, those could be eggs, what have you. The idea is you take a single diploid cell and you make 1, 2, 3, 4 haploid cells. You split the nucleus twice, and you make haploid cells as a result. Alright. So meiosis is a specialized type of cell division specifically for sexual reproduction. You take your chromosomes, you double them during the S phase of interphase, and then those are split twice to form these four, what we call gametes. Okay, these four gametes. What else do I want to say here? Okay, produce haploid gametes, haploid, single, 1N gametes, gametes. my kids are doing a music class in there, I can hear. Yeah, YouTube. Anyway, moving on. Gametes, egg and sperm are both gametes, can then fuse to fertilize the egg to form it's called a zygote. So you take an egg, you take a sperm, the sperm fertilizes the egg, that makes a complete set of chromosomes, 23 pairs, 23 from the egg, 23 from the sperm, 46 total, that is a zygote. So a zygote is that first cell that can then divide and become a human being. Alright, from the fertilized egg, that is a zygote. Sorry, all the all the music in the background. Let me just make sure I get this across to you. A egg is haploid, a sperm is haploid. If they come together, there's 23 chromosomes here, 23 chromosomes here, they form 23 pairs, that is a zygote. Yes. Okay, and then that zygote would divide mitotically to form a person. Perfect. That is perfect. Okay. Now, if events go wrong in meiosis anywhere along the way, you can end up with all sorts of problems, folks. And typically, this is going to be based off of having the wrong number of chromosomes in one of these eggs or sperm. Alright, this is a karyotype, and we're going to be talking about karyotypes a lot throughout this lecture. So I think it's worthy just to throw one up here now so you can see it. This is the non-human karyotype. You can tell that it's not a human karyotype because the chromosomal count. Do you see 23 pairs of chromosomes? No. Pair, up to 21. So this is a different animal altogether. But this is still a karyotype. What this is where we can map out where the chromosomes lie. Like this is the mom chromosome, this is the dad chromosome. You can see how they line up to see if there's any missing or see if there's any that are malformed or anything along those lines. Karyotypes are a way that we can take individual chromosomes, lay them out, and view them microscopically to see if there's any kind of problems with them. Alright, the importance of meiosis. Key points. 1. Meiosis introduces an enormous amount of diversity into populations. Sexual reproduction just really ramps up genetic variation. The number of offspring, hang on, do I have it on here? There are more than 70 trillion different genetic combinations possible from the mating of 2 humans. Let me say that again. 70 trillion different genetic combinations are possible. That's a massive, massive number. And I mean, it's pretty obvious. Like here are my kiddos, and I'm here to tell you. Okay, the 3 of them, they are all very different from one another. Very, very different. They come from my wife and I, but they are very, very different from one another for a reason, because there's so much crazy genetic variation that is possible. Like my son here's got my red hair, but he looks just like his mom in the face. It's a wild thing. Alright, and further, males and females do indeed differ in the way that they form gametes. We'll talk about that a little bit later on. It's a little more complicated. We'll get to that at the end. The idea is that male sperm production begins at puberty, so nothing beforehand, just at puberty, and then carries on till the end of life. And in females, you basically produce all your eggs early, pre-birth, and then once all the eggs are released that are made way on back, then you go through what's called menopause. And we'll talk more about that later. Perfect. Look at that cute little fellow. Oh my gosh, that's my youngest. Alright, so genetic variation. First things first, why sexual reproduction versus asexual reproduction? And I feel like I might have mentioned this a while back, but I could be mistaken, so let's do that now. Asexual reproduction is a wonderful idea when the environment is perfect for the organism. If I'm a bacterium and I'm hanging out in like the most perfect situation possible, all I need to do is reproduce myself asexually, make lots and lots and lots of copies of myself, and they're all going to be in the perfect condition as well. But as you probably realize, you don't always find yourself in the absolute perfect circumstance. And that's where having some genetic variability comes into play. When you have genetic variability, some organisms will have certain traits and other organisms will lack those traits. Sometimes the traits can be advantageous, that would be positive, a good thing, or sometimes the traits can be deleterious or a bad thing. The comment that I have here is on temperature rise due to climate change. If temperatures continue rising as a result of climate change, you can expect animals that have less fur. Like in a given species, like deer. Animals with less fur, and thus they would hold heat less easily, would be better adapted as opposed to organisms of the same species that has lots of fur. And you can probably ID with this. You know people that are hairy, and you know people that aren't hairy. So those are variations in a species, some of which could be advantageous and others of which could be deleterious. There's ways to look at this. A really nice example is this albino deer here. You can expect this organism not to be quite as successful as its compatriots here, because it is more likely to be killed by wolves or other predators. It's more visible. Yeah, perfect. That's perfect. So the idea is, let me just lay this on you in a little different way. One of the main tenets of evolution is that more organisms have to be born than can survive. So you produce lots of offspring. Think about like insects that produce 100 eggs at a time. They produce lots of offspring, and the ones that are best adapted are the ones that survive and pass on their genetics to the next generation. The concept would be that out of 100 born, maybe 5 or 10 will survive to reproduce and lay eggs, and that 5 or 10 are the best at living in whatever circumstance that is. We would call that, like Townsend would say, that nature is red in tooth and claw, that there is selective pressure upon these organisms, and having genetic variation means that you have traits that you could select for. Yeah. Alright, homologous chromosomes. So when you see, this is a human karyotype. When you see this human karyotype, and again you can tell it's human because you've got 1 through 22 what are called autosomes, and then a last set that are sex chromosomes. Both of these are X sex chromosomes, so that would be a female, gender-wise. These are, like one from mom, one from dad, called homologous chromosomes. Homologous, homologs, or homologous pairs, okay? And this is, again, one from mom, one from dad, one from mom, one from dad, one from mom, one from dad. So paternal homolog and what's called a maternal homolog. Now these are stained so that you can see the different traits that are found there. Basically, it's a long story, but you can see similar banding patterns. Why would these have such similar banding patterns? Because they are virtually identical to one another. your chromosome 1 is super similar to my chromosome 1 and super similar to anybody you see's chromosome 1. But there can be little bitty minor variations there, and those minor variations drive our own genetic variations. Alright, so they should look similar because they're all picking for the same basic genes. But there can be minor variations, and this is based off of a concept we refer to as alleles. Okay, we're going to get there in just a moment. So homologous chromosomes display genes. A gene, let me just put this in simple terminology. A gene is an area of a chromosome that codes for a protein. Like your genetic code, you're capable of making lots and lots and lots of different proteins. And a single gene is an area on a single chromosome that codes for a single protein. A protein comes from a gene on a chromosome. So, let's see. Genes occur at duplication, maternal copy, paternal copy, homologous chromosomes. We played this game. I want to get down to here, alleles. All right. There can be different versions of these genes that produce the most minor of tweaks on these proteins. And that's what gives us all of our crazy genetic variation. Okay, all of our wild genetic variation comes from us not having different proteins, but having different versions of those proteins. Our genes, we all have the same basic genetic setup to make the same basic proteins, but there can be minor variations on those proteins that are made. Our genes can have different alleles, different versions of the same gene. Variations that exist for a gene are called alleles. Like some of us can have genes for darker hair color. Some of us have genes for lighter hair color, red hair color, what have you. But it's all for hair color, okay? It's all for hair color. That's the way this works. So these are alleles, different alleles. Now, a single individual can be homozygous or heterozygous. Now, let's talk about what that means for a given trait. Let's talk about hair color. You could get the same allele for a gene from both mom and dad for light hair color, okay, as an example. It's not really a good example, but an example. You could get the same, like let's say hair color is found here and there. You get the exact same allele from mom as you get from dad. These variations of a gene, these alleles. The exact same allele from mom as dad. That would be considered homozygous for that trait. Identical alleles for a specific gene on both homologs are considered homozygous. You get the same thing from mom as you got from dad. Identical alleles, that is homozygous. Heterozygous, hetero means different. The idea is that mom gave you one allele for a trait, dad gave you a different allele for a trait. That makes you heterozygous for that trait. You have higher genetic variation as a result of this. Yeah, that's perfect. And I give you a little example here. Feel free to read it. But the idea is homozygous, same alleles, heterozygous, different alleles for a given single trait. All right. Okay, let's talk a little bit about sickle cell. Yeah, let's go ahead and do that. All right. Sickle cell anemia is a unique disorder. If you get a trait for sickle cell from your mom and a trait from sickle cell from your dad, in other words, you are homozygous for the trait for sickle cell, then you'll have all sorts of medical problems. Right? Very, very large, painful, unfun medical problems result from getting both a sickle cell trait from mom and a sickle cell trait from dad. So the kid's going to have a condition that's going to be an unfortunate situation. Minor technical difficulties. Back on task. Let me just make sure that we have this taken care of. Sickle cell is a good example of what's called an autosomal disorder. This is where one of your non-sex chromosomes, that would be 1 through 22 pairwise, is malfunctioned in a fashion that gives a trait for this disorder. Sickle cell is terrible. Is a terrible disorder where the blood cells don't form correctly, so they kind of pile up in certain areas of the body and they can't pass through the capillary beds well, and you end up with lack of oxygen and anoxic conditions. It's very painful. It hurts. You're very fatigued. It's a lot of problems. Sickle cell originated in sub-Saharan Africa, where it's actually a means of genetically defending the body against malaria. Okay, if you have a single sickle cell trait, you're basically immune to this terrible bloodborne pathogen called malaria. Whereas if you do not have a sickle cell trait, it's not unlikely that you'll die from malaria, especially like 100,000 years ago or so. So this was an adaptation in a certain part of the world to defend against malaria. But if you get a trait from mom and a trait from dad, and you're homozygous for sickle cell, you can end up with a whole variety of problems. The idea is, if you're heterozygous, then you are immune to malaria and healthy. Hemophilia is a good example of a sex-linked. That would be the 23rd chromosomal pair, would be that XX or XY in most circumstances, a sex-linked bleeding disorder. The idea is a person who has hemophilia, if they get a cut, instead of it drip, 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 clot, no more blood loss, instead of it being like that, the way you and I probably are, a person with hemophilia, if they get a cut, they can bleed to death from a small injury. Okay, very, very bad. All right, you basically can't clot blood. And this ran in high numbers in the European royal family. The European royal family, if you're not aware, is horribly inbred, and you end up with a circumstance called inbreeding depression in this particular case. Basically, when you breed together cousins for 100 or 200 years or more in this case, you end up putting together lots of genetically deleterious traits, and you end up with relatively sick people. And this child is one of those. So this is the son of Alexandra and Nicholas II of Russia. I think they were first cousins. And Alexei had hemophilia. Okay, and what would happen here is the genetics for blood clotting travel only on the X chromosome. So ladies, as you can see here, the females get 2 X chromosomes. Ergo, they get 2 chances to get the appropriate genetics for blood clotting. Whereas a male only gets a single X chromosome. Like Alexei here, only got 1 X chromosome, and it happened to be a hemophilia chromosome from his mom. So he ends up with major, major problems that had to be addressed so that they could continue this line of czars in Russia. To deal with this, they brought in a faith healer named Rasputin. And if you want to read a wild story, I invite you to go read about Alexei, Alexandra, and Rasputin. Their relationship was quite fascinating. And pretty wild. So go give it a read if you'd like. It's pretty neat. Alright. Yes, this is where the Disney movie came from. Let's go here. So, meiosis is reduction division, I should say. I'm not going to go into a lot of detail here, especially considering I've already taught this once and the freaking memory card was full. What I will say is this. In meiosis, what you do is you double your chromosomal count. In the meiosis 1, that splits into 2 identical cells, and then those identical cells will further split in meiosis 2 to produce 4 what we call haploid cells. How many times do I have to say this, right? You take a diploid cell, you put it through meiosis 1, meiosis 2, this is basically 2 mitotic divisions. They're meiotic, but you get my point, and produce 4 cells, because these could be sperm, these could be eggs, but the idea is they are haploid. They are 1N. They are haploid cells. They are gametes, as it were. Yeah. Same thing here. So the idea is, here's meiosis 1, produces a couple of cells with a double chromosomal count, and then those will split further to produce 4 haploid cells at the end of the day, be those sperm or eggs. And here is a nice animation of this. The idea is in mitosis, you produce 2 identical cells with the same chromosomal count as mom and dad, and then in meiosis, you produce haploid cells with half the chromosomal count of mom and dad. This is very important. You'll be seeing this again. Alright, now, I foresee on your test a question set where A is mitosis and B is meiosis, and you have to go through and tell me what's what. It's very simple. Let's just run through all of this together. Requires a single nuclear division. That's obviously mitosis. Remember, in meiosis, there's meiosis 1 and meiosis 2. That's 2 nuclear divisions. Mitosis. Chromosomes do not synapse or crossover, whereas in meiosis, chromosomes do synapse and crossover. And that's literally the next conversation we're going to have. Synapsing, crossing over, this is going to be one of the ways that sexual reproduction drives crazy amounts of genetic variation. The chromosomes can come together and change each other, basically. And we'll talk about how that works in mere moments. Let's see. Centromeres, yeah. Chromosomal count is preserved making 2 end cells or diploid cells in mitosis. In meiosis, you make 4 haploid cells. In mitosis, you make 2 daughter nuclei. In meiosis, there are 4 produced, be those sperm or eggs. In mitosis, the daughter cells are genetically identical to one another. In meiosis, the product daughter cells are not genetically the same. And then in mitosis, we use mitosis for asexual reproduction and growth. Okay, like you grow taller because of mitosis. Whereas in meiosis, it's used only for sexual reproduction. That is a fact. Alright, so let's talk about crazy amounts of genetic variation. So here's a mom and her offspring. You can guarantee that they're all a little bit different from one another. Genetic variation is very important for the environment. You must have high levels of genetic variation in a population for them to survive in the wild. It's very important. We have seen in many cases where you'll have some kind of crazy monoculture where all the organisms are very similar, and then disease outbreaks and just kills them all. Okay, it's a real thing. Okay, so asexual organisms do have some mutations that leads to them having a little bit of genetic variation, but they can't even touch what happens via genetic variation in meiotically dividing, sexually reproducing organisms. And that's because meiosis allows for 2 processes called crossing over and independent assortment. And let's talk about those. Okay, crossing over. What will happen is in meiosis, let's see, I think it's, where does it happen? What part of meiosis does it happen in? I can't remember, and it's certainly not on my mind right now, so let's just ignore that as a concept. Let's say the following. So, in meiosis, as the nuclei are doubling their chromosomal content and then dividing, what can happen is you'll form up in between sister chromatids these synaptonemal complexes, basically where the 2 chromosomes come together and they just twist and wrap all around one another. This is a real synaptonemal complex in a scanning electron micrograph. They twist upon one another and they twist, and little bits can snap off and then recombine to different chromosomes. So these 2 can come together, and bits of an allele from here can break off and attach over here, and bits from here can break off and attach over here. And what you end up with as a result of crossing over is chromosomes which are no longer identical to the chromosomes that mom and dad gave. Alright? So this chromosome is nothing like anything dad has. It doesn't represent either his maternal or paternal chromosome. This is a different chromosome now. It's completely unique. And so is this one. So the idea is that through crossing over, you end up with crazy variation in the individual chromosomes. They are now quite different from the chromosomes that Dad originally had. The chromosomes that come out as sperm are very unique. And the same thing with eggs from Mom. Mom's chromosomes are no longer identical to the chromosomes in the eggs. Because of crossing over, they can be very unique. You end up with very unique chromosomal output. Nothing is completely new here, but the daughter cells are more unique. So this would not represent either of the chromosomes found in mom or dad for that specific locus. It's different now. It's different. Yeah, that's good enough. Good enough. Main point here, crossing over dramatically increases genetic variation. Yes. And then there's independent assortment. So imagine we have an organism with 3 chromosomes total. All right, 1, 2, 3 chromosomes total. 3 chromosomes total, as seen here. 1, 2, 3. The idea is that if for chromosome 1, if Dad's making sperm, this one goes into the first sperm. There is no telling what's going to go into the next one. The next 2 and 3 here, they assort themselves independently. So when you're producing eggs, producing sperm, you can end up just with 3 chromosomes with a wild number of combinations of these chromosomes, because where one of them goes has no impact over where the next ones go. The chromosomes assort themselves independently. One doesn't drag the other. You can't end up with, well, you can, but it's highly unlikely to end up with a bunch of identical gamete cells, because they're going to have crazy independent assortment that results in a random mixing of alleles and gametes, okay? Yeah, they assort themselves independently. That's the way this goes. Now, as a result of this, when you take two gametes and you fertilize, or let me rephrase, when you take a egg and you fertilize that with a sperm, when you take two gametes and they come together to form a diploid zygote, you can end up with wild numbers of variation. So just from independent assortment, you can end up with this many different organisms. This many different organisms, just from independent assortment. When you add in just one event of crossing over, you can end up with this many different zygotes. So you look at me and the kids, right? Me and the wife and the kids, we're all a little different from one another. Okay, we're all a little different from one another because of independent assortment, because of crossing over. With just one crossing over event, you can get this many different zygotes. And the thing is that crossing over happens multiple times. Okay, multiple times. So you can end up with just ridiculous numbers of different genetic variations possible from sexual reproduction. And for humans, this is less of a thing because we have small numbers of offspring. But again, think about frogs. Frogs can breed multiple times a year, producing hundreds and hundreds and hundreds and hundreds of offspring, all of which can have completely unique genetic codes. And this is going to drive genetic variation, which increases the likelihood of their survival as a species. Because of this increased genetic variation, the population as a whole is more healthy. Alright, perfect. And you sometimes end up with weird things. I just find this kind of fascinating. What you see here is me as a young man, and this is my uncle, or great uncle, I guess, J.E. This was my grandmother's brother. He was a paratrooper back in the day, and boy, people say we look quite similar. Okay, same jaw, same lips, same nose. There's a lot there. It's always been said, we look kind of the same. So it's kind of neat. Sometimes you can end up with unique variations that are passed down through populations. Alright, parthenogenesis. Just for fun, kind of throwing this in there, there can be some other weird things that can happen, I should say. Organisms that normally reproduce sexually, there are some cases where they can reproduce asexually. Given certain circumstances, they can't find mates, or if the environment is ideal, they are capable of reproducing asexually and producing offspring without fertilized eggs. Parthenogenesis, this can be done. And plants can do something similar called alternation of generations. Algae, ferns do this. So what can happen is sometimes they can reproduce in haploid fashion, producing what they call gametophytes, and sometimes they can reproduce in diploid fashion, called sporophytes. It's kind of a long story, and I'm not that interested in getting into it, to be honest. But the idea is that you can have some generations that are haploid, some generations that are diploid. Just depends on what the environment needs and how the organism reproduces. Good enough for me. I'm not that concerned. What I want to get to is here. I'm going to restart the video. Alright, so hormones produced by the testis and ovaries. Okay, so the testis are kind of famous for producing testosterone, whereas the ovaries are kind of famous for producing estrogen and progesterone. And what these are going to do is they're going to drive male and female secondary sexual characteristics. So there's all sorts of variations that occur as a result of these hormones. When you are producing them, or if you're not producing them, it can drive all sorts of variations in the body, even in utero. So what you see here is a male and female, and I honestly can't remember which one's which. I want to say that's the male and that's the female. But what you can see is that in utero, we're almost identical. It's the hormones that drive the development of our sex organs, that drive the way that we develop through puberty, that drive our variations in our physical characteristics and the way that we are built. The hormones are incredibly important to this. Incredibly important. And let's see, what do I want to say here? I really need to add some stuff into this, but the idea is this. This is going to be a little more complicated, but gametogenesis is broken down into what we call spermatogenesis, the formation of sperm, and oogenesis, or oogenesis. I tend to say oogenesis. which is the formation of eggs. And what I want to say, what you need to know, okay, this is the key concept. This is something that we've been tiptoeing around this entire lecture, is that in spermatogenesis, when you take a single diploid cell and you make 4 haploid cells, all 4 haploid cells become sperm. Okay, all 4 haploid cells become sperm. In oogenesis, what you do is you take 1 diploid cell and you make 4 haploid nuclei, only one of which, only one becomes an egg or an ova, okay? I'm sure I have it in here someplace. But anyway, only one of the 4 develops into a egg that can then be fertilized to produce a person. And the reason for this is very simple, and I really kind of had this lecture out of alignment, but the idea is, in sperm, what you want from a sperm is for them to shed all of their cytoplasm, all of their cellular components, and basically just be a head of DNA and a tail to swim with. That's why sperm don't live very long. They have a very short shelf life. Whereas an egg, what you want to do is conserve all the cytoplasm. Conserve all the cytoplasm into a single egg, shed the polar bodies, is what these are called. Polar bodies are the spare nuclei from the meiotic division. You shed 1, 2, 3 nuclei. You keep all the cytoplasm in a single cell with a single nucleus containing a haploid chromosomal count. Then sperm can meet that egg, fertilization happens, and you can produce a zygote. So that's how this works, okay? Key point. Spermatogenesis, you make 4 sperm. Oogenesis, you make 1 functional egg. 1 functional egg from oogenesis. Perfect. And just for fun, I throw this up here. We are not that different from other organisms. So here's a plant, and I'm not going to go into the details of reproduction for you. I trust that we're all kind of familiar with this. But the idea is that if sperm get into the uterus of a female and an egg is released from that ovary... My Alexa is going crazy in there. Anyway, if sperm get into the uterus and an egg is released from that ovary, you can get fertilization and implantation and produce a zygote that can then become another person. In plants, it's quite similar. Instead of testes, they have anthers, and those anthers produce what is in essence sperm. That sperm can be deposited on this sticky upper portion called a stigma, where it can migrate its way down in the same fashion as sperm migrate their way up, get into what is in essence an ovary, and fertilize an egg in the same basic way. So plants and animals are very similar in the way they conduct sexual reproduction. You can even have, like, certain plants produce male and female plants. Like, I think, is it ginseng? I think ginseng plants produce male and female plants. So the female plants will actually produce an odor that elicits the male plants to start producing pollen, which is in essence sperm, to fertilize the female plants. It's a wild thing. So, yeah, plants and animals, quite similar in reality. Alright, let's see. Life cycle of humans. Sperm and egg are produced in meiosis and spermatogenesis and oogenesis. Sperm and egg fuse at fertilization, resulting in a zygote. So these are gametes. These are gametes, gamete. They fuse to form a zygote. That zygote, oh, okay, I see where we're going. That zygote's going to divide mitotically to form a human, which is going to be 2N, 2N, 2N. Full-grown humans can then reproduce, functioning to produce egg and sperm. They can then fuse. These are going to be haploid, 1N, can fuse. That is fertilization to form a zygote, which is 2N. It is cyclic. Okay, it is quite cyclic. Perfect. Perfect. Yeah, I'm pretty happy with that. I'm not terribly concerned. These are some neat pictures. So what you see here is the only time it's ever been documented of an egg being released from an ovary. I feel like this was in Europe, and for years and years, like, I've got some funny books that are really old, reproductive history books in my office. I normally would bring these into class and show you. It's pretty funny, where they describe the release of an egg from a ovary as an explosion event. Like, it just bursts out this egg and shoots it through the fallopian tube, which is complete nonsense, alright? Again, people in the 50s just had no idea. This is the first time it was ever documented. This was just a few years ago, and again, I believe it was in Europe, where a Graafian follicle, which is what this is an ovary, this is a Graafian follicle, it's going to secrete an egg, and it slowly lets the egg loose, and then the egg is collected by fimbriae in the uterine tubes and transmitted towards the uterus. This is real sperm coming down and attacking an egg, basically. So what they have on their heads is an enzyme that breaks down the egg's external coating, and the moment that one of the sperm breaks through the egg's external coating, it causes a chain reaction, a chemical change that makes the egg's coating completely impervious. So only one sperm ever gets in. At least that's the goal. There are some weird variations, but neither here nor there. But that's how fertilization takes place. This down here is a tiny little suction device. This is a human, or any egg cell for that matter. This is an egg cell, and what they've done is they've used a syringe to suck out all the nuclear material from that cell, and they can then go back in and insert a nucleus of whatever the heck they want to insert. And grow organisms based off of this fashion. They can even suck out the nucleus, edit the DNA, and then put that nucleus back in. And I invite you to go and read about what they call designer babies. Today, right now, it's very easy to go to a expensive fertility clinic and guarantee if you're going to have a boy or a girl. Very simple. Very simple. And there is some evidence out there that in some other countries, you can have them go in and edit the DNA. So they can go in and look at the genetic code that is produced from multiple zygotes. And so make sure that they're not going to have certain medical conditions. If you want to have blonde hair versus brown hair, they can go in and do that. It's possible. It's very unethical, and there's not a lot of data out there saying that it's happening with regularity. There are people who have claimed to have done it, and that was last year, so I predict it's gone up since then. So yeah, there's some wild stuff, man. Some pretty wild stuff out there. Alright, this is basically just going through what I've already said. Spermatogonial cells, an initial sperm cell, or spermatogonial cell that is diploid, can divide meiotically to produce 1, 2, 3, 4 haploid sperm. And the idea is that these sperm have shed all their cytoplasm. They shed their cytoplasm, they become very small and very streamlined, and they swim very fast towards that egg. They display what's called positive chemotaxis. They can sense from which uterine tube the egg has arisen, and they'll all travel to the egg. It's a wild thing. What else do I want to say here? Oh, quite fascinatingly, it's projected that male sperm, because they contain a Y chromosome, which is much smaller than an X chromosome, tend to be much faster. So if your egg is released the moment at copulation, the chance of having a male child is vastly increased. Whereas if the egg is released a little late, the male sperm tend to die very fast, and the female sperm, which are bigger and heavier, they tend to live a little longer. So if the egg is released late, then the fertilization occurs and you tend to have a female child. So it's kind of when copulation occurs as to if you're going to have a male or female. Now there's obviously variation in that, but that's an important bit to think about, which is kind of neat. It's a neat thing. Right. Okay, so last comment. So it takes 5 days for spermatogenesis to occur, and again, you shed cytoplasm, whereas in oogenesis, what you're making is 1, 2, 3 what are called polar bodies that are shed, non-fertilized, can't be fertilized, they're useless for us. But the single egg that contains all the cytoplasm at the end of the day, that's the one that can be fertilized through oogenesis. And it's actually a little more complicated than that. I invite you to read this, but I am not going to test you on those complications. I simply want you to realize that you take a single diploid cell, you make 4 haploid nuclei from this, but only 1 functional egg cell that can then be fertilized as a result of oogenesis. Perfect. Changes in chromosome numbers. So there are 2 terms to describe here. The first one is euploidy. Euploidy is where you have the appropriate number of chromosomes. And then there's an aneuploidy event. Aneuploidies are any time that you have an abnormal chromosomal count. These can be monosomy events where you have a single chromosome at a locus, or trisomy events where you have more than 2 chromosomes at a locus, 3 trisomy. So let's say we're going to have an aneuploidy. You take a single diploid cell, you divide it mitotically, but look, there's a non-disjunction. Non-disjunction events are where there's a failure of the chromosomes to separate correctly. So you have a non-disjunction event where instead of having 1 chromosome go here and 1 chromosome go here, they both go here. And then as you divide meiotically, you end up with 1, 2, 3 chromosomes. Okay? 1, 2, 3 chromosomes. Well, you get my point. More than the appropriate number of chromosomes. as a result of fertilization. It is a little bit more complicated. You got to think about this as egg and sperm. But the idea is that anytime there's a non-disjunction event where the chromosomes don't split the way they're supposed to split, you end up with problems. Okay, you end up with problems. Sometimes it's just a trisomy, which is more easily tolerated by the body. But if it's more than that, sometimes you can end up with 4, 5, 6 copies of a gene in a single egg cell, and that's not ideal. Or 4, 5, 6 copies of a chromosome. Yeah, non-destruction events. Monosomy, trisomy, both of these is aneuploidies, and then here is 1, 2, that is euploidy, or the correct number. Now, the most common trisomy is trisomy 21. This results in Down syndrome. Again, the only problem with Down syndrome is it's a genetic abnormality where there is a third 21st chromosome. That third 21st chromosome results in Down syndrome and all the conditions that we associate with Down syndrome. So even just a minor change, one extra chromosome out of 23 pairs can result in a lot of physical problems. Okay, a lot of physical problems. Trisomy 21, an aneuploidy. Okay, that is an aneuploidy, a result of a non-disjunction event. Let's see. Oh, and then there's some kind of fascinating ones. Other aneuploidies that deal primarily with the sex chromosomes. So you know that a female is XX, a male is XY, but there can be extra or missing copies of these. A good example of this is Turner syndrome. So a female with Turner syndrome gets an X chromosome from mom, but nothing from dad. Or alternatively, an X chromosome from dad, but nothing from mom in terms of the sex chromosomes. So you have 1 through 22 autosomes, and then sex chromosomes, you just have 1 X. Okay, 1 X. That is Turner syndrome. Shorter individuals, broad chest, widely spaced nipples, normal intelligence most of the time, and basically with hormone therapy, mostly normal. Okay, mostly normal. But with a missing chromosome here. We tend to find that sex chromosome malformations are more easily tolerated. You can look up images of this. It's pretty fascinating. And then there's Klinefelter syndrome. Klinefelter syndrome is seen in males where you have more than 1 X chromosome. So imagine okay, let me first say this. If you got a Y chromosome, you're male. Okay, you're going to have male parts. That's just how this works. There's no such thing as having a little bit of both. That's not the way this works. You're either going to have male parts or female parts. They could be degenerated to virtually nothing, but they are either going to be male parts or female parts. And Klinefelter syndrome is fascinating. Amongst these, what you have here is 2 X chromosomes. So mom gives 2 X chromosomes, or dad gives an X and a Y, some way, shape, or form, and you get fertilization, and you end up with XXY, or sometimes XXX, many X's and a Y. The more extra chromosomes you have, the more problems you end up with. It's not uncommon for people with 2 X chromosomes, XXY male, Klinefelter's male, to not even really notice it. Okay, to not even really notice it. But the idea is these will have underdeveloped testes and prostate, typical breast development to some degree, kind of variations in the arms and legs and hands, but typically mostly normal. Okay, mostly normal. Unless, again, you have lots of X chromosomes, in which case you can end up with problems. Now, this is not to be mistaken with what's called SRY gene deletion. What you have here is an individual that's born XY chromosome, but they develop completely and utterly female. In every way, they are female. All right, they are female because they lack a gene that basically allows them to respond to androgen. It's an androgen insensitivity. Lack of what we call an SRY gene, or testis determining factor, which plays a role in male development. Okay, male development. And this became a thing in the it's 96 Olympics, okay, in Atlanta. To test to make sure there weren't males competing as females. How things have changed. To make sure that there weren't males competing as females, what they did was they tested them for an SRY gene. Okay, pretty simple, straightforward test. Tested them for SRY, and it turns out that there were several females. I think it's 8. Yeah, 8 female participants tested positive for an SRY gene, one of which had metalled. And they were going to strip away the metal. They were like, oh, you're male and you're competing as female. They ran some genetic tests, and in fact, this person did have an X and a Y chromosome, but they were androgen insensitive. In other words, they developed as female. And so because they developed this female, even though they had an X and a Y chromosome, they considered this person to be female, and thus they kept their metal, which is pretty fascinating. Genetics. Genetics. It's all about genetics. This is an amazing, amazing set of instructions for making people here. And I hope that you have enjoyed this. If any of this is fascinating to you, I would invite you to look it up. It's pretty wild how all this stuff fits together. Boy, is it fascinating. Right? Boy, is it fascinating. Go and do some reading. All right? Go and do some reading. It's pretty neat stuff. And I'll be sending you quizzes post haste. Thanks.